So we do have evidence for specific coding. In other words, we do have evidence that shows we have specific structures in our taste system for specific tastes. But we also have evidence for distributed coding, this sort of mixture of different flavors, creating different sensations. Some researchers are thinking that the neural system for taste might be like the visual system for color. Do you remember that we had the trichromatic theories of color vision, where we had where we were mixing the different cones that could perceive different wavelengths of light, and presumably somehow, for example, through the opponent process method, those different cones receive different wavelengths of light, and that information is combined to create our perception of color. Some researchers are thinking that taste might work the same way, where these different chemicals sort of combine to give us this perception of taste. We really don't have a good explanation right now for ultimately how we ultimately perceive taste. We know a lot about how the sense mechanism works and the chemistry and the physiology, but ultimately we really don't have a good explanation for how all of that hardware and plumbing works together to create our perceptions of taste. Of course, there's great individual differences in taste. There are tremendous individual differences in taste, which of course everybody knows. You know, we know, for example, that some people really like bitter, foods. Some people like really sweet foods. We have very strong individual differences in taste. We're able to detect these with these chemicals such as PTC and PROP, right? PTC and PROP. PTC is the bitter one, and we have people that can taste these chemicals, not taste these chemicals, and are really sensitive or can really taste these chemicals. So tasters, non-tasters, and super tasters. One of the things that we found is that the tasters have more taste buds than the non-tasters. So in other words, if you can taste the bitterness in the PTC, and then another person doesn't taste the bitterness in the PTC, we have found that you might have more taste buds for bitter than that other person does, which is really interesting. The tasters seem to have specialized receptors for these compounds. So the more you can taste these compounds, the more receptors you have in your tongue that are specialized for detecting these compounds, which is really interesting. The super tasters can appear to be more sensitive to bitter substances than tasters. They can really taste bitter substances because they seem to have a lot more receptors than the tasters do, and certainly than the non-tasters do. Here's a picture of concentrations of taste buds and papillae in A and B. Notice that and this is from the area of the tongue that contains lots of bitter receptors. Notice that there's lots more taste buds located in A than there are in B. So you can actually see physical differences in these folks that have differences in taste perception, and these physical differences reflect also perceptual differences as well. So let's now switch to the next topic. We've talked about taste. Now let's talk about olfaction or smell, the olfaction system. Many animals are macrosmatic. They have a very keen sense of smell. So, for example, dogs, right? Dogs and rats. They have very strong senses of smell because they need it for survival. Humans are microsmatic. We do not have a strong sense of smell as other critters do because it's not so crucial to our survival. However, we do respond to, at least at a subconscious level, we do respond to certain smells. For example, Singh and Bronstat, this is Devendra Singh and Bronstat, showed a relationship between a men's rating of women's body odors and the women's menstrual cycle, so that there was this weak relationship where men could tell where women were in their menstrual cycle based on their body odor. You see a lot of these data where we see, under certain conditions, we can respond to things like pheromones and whatnot. So we do have certain abilities, but we're certainly not like some of the other critters, like dogs, for example. Dogs have extremely keen senses of smell. How do we measure odor detection? Well, you have to remember, first of all, there's a lot of techniques for doing this that have been around for a very long time, and it should be obvious to you why that is. Because there have been a lot of people interested over the years in, of course, producing good smells. Perfume makers and soap makers and all these other things. So we have a lot of techniques for detecting smells. So how do you detect smells? Well, the simplest way is with the yes/no procedure, right? So you give them an odor, and then you give them the same thing, like a rag or whatever, that doesn't have the odor, and they say, yes, I smell it, or no, I don't. The problem with the yes/no procedure, sometimes you get a lot of false positives, right? It kind of results in a lot of false positives where they will say they smell something, but they don't actually. Another one is that's a little bit better, is the force choice procedure. And what you do here is 2 trials are given. Of course, you counterbalance it. One trial has the odor and the other one doesn't have the odor. And there's different concentrations of the odor, right? So it's real weak and real strong. And so you give the odor, and then you don't give the odor. And then you ask the participant, okay, which one of these 2 trials smells the strongest? And if they can't tell a difference, that means that they can't detect it. But when they start smelling, when they can detect it, then that's when you can tell where the threshold is. Doing these types of studies, we've learned that rats are 8 to 50 times more sensitive to odors than humans. However, if you think that's impressive, dogs are 300 to 10,000 times more sensitive to odors than humans are, which is amazing, which is why dogs, of course, are used for security and law enforcement to detect drugs and chemicals and explosives and all these things. They have extremely sensitive smells. That's why dogs like to stick their heads out the window when they drive, because dogs map out their world according to smells, and when they have their heads out the window, they just get this huge waft of smells, and they just sort of enjoy for them, that's looking at the scenery, right? It's just getting those smells, right? What's interesting is that it's not the receptors. So in other words, it's not like that dogs, for example, have super duper sensitive receptors. Rather, the big difference is in the number of receptors. They have the same receptors as we do, pretty much, but they just have a lot more of them. So if you measure all of the olfactory receptors that we have in our noses, up in our mucosa, humans have an average of 10 million receptor cells. Dogs have over 1 billion receptor cells. So they have an order of magnitudes more receptor cells than we do. And this is what gives them their extremely keen sense of smell. So in measuring the difference threshold, right, the difference threshold is the smallest difference in concentration that you can detect between 2 samples. So you have 2 samples, and you slowly increase the concentration until you can measure that one smells different than the other one. They have devices that they've created called olfactometers, which is basically it's this air device where these hoses are stuck up in your nose, and they like snort smells up into your nose, and then you're supposed to tell them if you can smell a difference or not. and it's roughly 10%, 10 or 11% of a concentration where you can detect a change in the concentration. So let's say I gave you a smell of something sweet, and then I give you a forced choice where I keep increasing the concentration. Well, you're going to tell me that one is sweeter than the other. You're going to tell me that they're both the same until I hit 11% concentration difference. When I hit that 11%, you're going to say, oh yeah, that one smells sweeter than that one does because it's 11% more. So this chart here gives us some odor detection thresholds in terms of parts per billion. So how many parts of this molecule has to be in per billion parts of air in order for us to detect it. So the higher the number you see in the odor threshold, the less sensitive we are to it. So methanol, methanol and acetone, which are both organic compounds. For methanol, it takes 141,000 parts per billion before we're able to detect it in the air. Methanol is a type of alcohol. It's not particularly dangerous if you smell it. So there's no reason we should be too sensitive to it. Acetone, another naturally occurring organic compound, 15,000 parts per billion. So we're more sensitive to that. Formaldehyde, which is a fairly dangerous chemical, right? If you eat too much of that, you'll die. It only takes 870 parts per billion for us to detect its presence. Menthol. Menthol is the chemical that gives mint its distinctive smell. So menthol is produced by mint plants. Right? So it's a very powerful thing. We're very sensitive to menthol. As you can see here, it only takes 40 parts per billion before that we can detect. And then finally, a chemical called T-butyl mercaptan. This is the chemical that's used that they put into natural gas so that you can smell it if it escapes. Because although natural gas is odorless, so if your house gets filled up with natural gas, you would not know it until you pass out. So they put this chemical into the natural gas so that you can smell it. So if you've ever smelled natural gas, you've smelled this stuff. We are very sensitive to this compound. It only takes 0.3 parts per billion, or 1 part per 3 billion. For us to detect it, so which is good, right? You want to be able to be sensitive to that stuff because we use it to mark the presence of natural gas, which could be dangerous for you. So how do we go about identifying odors, what you smell? So the recognition threshold, of course, is the concentration needed to determine the quality of an odorant. So you need a certain amount of it to be able to detect what it is, all right? We can discriminate among 100,000 odors, so I can tell you if one odor is different than the other, but we have a lot of trouble labeling them. I can say, yeah, that's different than that, but I can't really say what it is. And it's not because we're not sensitive. We can detect the changes, but it's hard for whatever reason. It's very difficult for us to map the language of what something is called to the particular smell. We just have a lot of trouble recalling a word, the term for a particular odor from memory. So this is an olfactometer, the thing I was telling you about before. There's compressed air. And valves that control the flow of compressed air. So what they'll do is they'll give you a blast of, say, for example, just regular air, and then the odorant, and then they'll use the valves to control the flow of the air, and then they'll blow out the odorant out of the system through the ventilation tube, then they'll give you another blast of something else. This is a very simple drawing. These devices are actually fairly complex, and the nice ones are computer controlled now, but this is essentially the way an olfactometer works, and the way that odor researchers get their data. This is what people that make laundry detergents and perfumes and and all the other things that we smell in our daily lives. This is how they do the research to determine how we're perceiving different smells.